past few decades, stem cell therapy has revolutionized the field of medicine. Since the 1980s, scientists have been exploring the potential of stem cells in neurodegenerative treatment and prevention. Such research offers hope for a wide range of disorders, but today we'll focus on its therapeutic applications in diseases such as Parkinson's and ALS. The mechanism underlying all forms of neurodegenerative disease is the progressive loss of neuron structure and function, often leading to complete neuron death. And unfortunately, there are no pharmacological or neurosurgical treatment options currently available that effectively arrest such neurodegeneration. Stem cell therapy, on the other hand, shows promise in its potential to regenerate certain types of neural tissue, thereby ameliorating neurodegeneration occurring at different levels of the neuronal circuitry. A stem cell is an undifferentiated or pluripotent cell, meaning it is capable of producing identical daughter cells. Stem cells and their progeny will mimic the cells of any given location, differentiating into specialized cells such as muscle or red blood cells, which hold significant therapeutic value as these cells have the potential to serve as an internal repair system in many tissues, dividing essentially without limit to replenish other cells for as long as the organism is alive. In the case of the nervous system, stem cell differentiation or neurogenesis produces neurons, astrocytes, and oligodendrocytes, and, contrary to popular belief, this process continues into adulthood. Neurogenesis has been established in the adult brains of rodents and macaques localized to the subventricular zone and regions of the hippocampus. In adult humans, neurogenesis has also been identified in hippocampal regions but has not yet been established in the subventricular zone. Furthermore, disturbances to neurogenesis, the compromisation of this regenerative ability, have been implicated in cases of neurodegenerative disease and injury, suggesting that stem cell treatments which re-establish neurogenesis have therapeutic potential. Research into this area has identified two types of stem cells with the greatest therapeutic potential: embryonic and mesenchymal stem cells. Embryonic stem cells are derived from embryos of artificially fertilized eggs and are capable of differentiating into all germ layers, i.e., any type of cell. On the other hand, mesenchymal stem cells are sourced from umbilical cord blood and have a migratory capacity similar to that of neural stem cells, increasing the distribution of cell regeneration and therefore therapeutic potential. Neurodegenerative diseases such as Parkinson's are another major focus of ongoing research into stem cell use in neuroscience. Parkinson's disease affects the basal ganglia and is characterized by progressive degeneration of dopaminergic neurons, that is, neurons responsible for the secretion of dopamine. People suffering Parkinson's disease most commonly experience a resting tremor, slowness of movement, rigidity in the body, and depreciated postural reflexes. Clinical trials in this area have involved giving dopamine neuron progenitors to patients suffering Parkinson's disease. However, this method is controversial, bringing up many ethical concerns, as the cells themselves are sourced from fetal brain tissue. Consequently, researchers are looking into finding an ethically sound cell source, as well as exploring the option of reprogramming damaged adult cells into induced pluripotent stem cells, which is currently the most promising approach, although clinical trials have yet to be conducted. ALS is another neurological disease where stem cell therapies offer hope. It is an adult-onset disease characterized by the death of upper and lower motor neurons, so motor neurons in the cortex, brainstem, and spinal cord, with subsequent muscular paralysis and atrophy leading to respiratory difficulties and eventual death. Diagnosis is generally between 50 and 60 years of age, with an average lifespan of 3 to 4 years. There is no known cure for this disease, and the only approved treatment extends survival a few months. Compared to other neurodegenerative diseases, ALS presents certain challenges in treatment via stem cell therapy, such as an unknown pathogenesis and a lack of knowledge about the spread and progression of the disease. These factors make it difficult to determine the ideal site for stem cell implantation. Theoretically, the objective of ALS stem cell therapy would be the replacement or substitution of motor neurons, but current strategies are also looking at regulating inflammation and the expression of neurotrophic factors, which are molecules that support neuron growth, survival, and differentiation. A recent study performed in transgenic rats showed improved motor function following treatment with modified stem cells. The cells were modified to release GDNF, a protein integral to neuron survival. However, the findings of this study were limited by the rat's lifespan, meaning no long-term effects were recorded. The strict internal regulation of the central nervous system is another factor hampering the progression of stem cell-based therapies. In the case of ALS, further, time-consuming studies have to be performed to determine the safety of implanting neural stem cells into the spinal cord or frontal motor cortex. To date, such studies have revealed that 90% of motor cortex implantation patients survived for at least a year after surgery, and spinal cord implantation has been well tolerated with minimal short or long-term complications. Moving forward, research aims to identify the most effective progenitor cell as well as the safest and least invasive route of administration for each neurodegenerative disease. While we still have a way to go, such research is worthwhile as stem cell therapies offer hope to those suffering from currently incurable neurodegenerative diseases.
While these results are promising, more work is required to optimize treatment delivery and refine the stem cell therapies themselves. The concept of neurorestoration has evolved rapidly over the last decade to the point where a recent study identified up to 106 of either ongoing or planned neurorestorative clinical trials. These stem cell therapies have set off both interest and alarms in the scientific community, but despite copious studies, there are still more questions than answers. One reason for this is the vast majority of studies are conducted in animal models, meaning there is a paucity of data on clinical outcomes and long-term safety of neural stem cell therapies in humans.